Welcome students. Today I'm going to explain the basic characters of seed dormancy and germination, how they act, what are the crucial factors which can regulate dormancy and germination, how we can break dormancy, all these things. So firstly, what actually germination is? Germination is the beginning of growth and development of the dormant embryo. And if we consider dormancy, it is a temporary suspension of growth of the plant tissue and organ, and these two are very important aspects of the plant physiology and development. Firstly, there are some factors or regulators which can control the germination process. And if we want to just list them, these are the most important one, like gibberellins, cytokinins, ethylene, abscisic acid, and light. There are also some other factors also, but these are most crucial. Firstly, I want to mention about gibberellins. These can stimulate germination in both dormant and non-dormant seeds. This stimulatory or promotive effect is specially observed in seeds where dormancy is imposed by various mechanisms like incomplete embryo development, mechanically resistant seed coat, etc. Generally, the seeds require dry storage condition for 3 to 5 months at root temperature and which allow maximum germination. It means that in these seeds, the dry storage help the embryo to acquire the capacity to produce gibberellic acid. During germination, the amount of sugars which is present in the endosperm control the gibberellic acid synthesis in the embryo. Gibberellins at high temperature can break the dormancy of positively photoblastic seeds, like Lactuca species, and some other negatively photoblastic seeds also, and some non-photoblastic seeds which require stratification. All the above types of seeds germinate under non-inductive condition when treated with gibberellins. And previously we also have a clear idea about the basic role of gibberellin, that they have a role on flowering, they have a role on vernalization, and they also help in the process of internodal elongation. Now, cytokinins or kinetin. This also can induce germination in positively photosensitive seeds like lettuce. And actually, the naturally occurring cytokinins, which usually found in the tRNA or transfer RNAs, located near the anticodon. When cytokines are present in small amount, tRNAs would be deficient and protein synthesis depressed. So by exogenously applied cytokinin, protein synthesis is enhanced and thus affecting the quick germination. So in this way, cytokine can induce the seed germination. Now ethylene, which help in the ripening process, we all know. Ethylene promotes the seed germination also. Ethylene is produced by germinating seeds, and it is related to the number of individual seeds germination. Now, abscisic acid. The abscisic acid inhibits certain enzymes associated with germination, like endo-beta mannase in lettuce. But it has been shown that the reduced enzyme activity is due to reduced rate of germination. ABA also inhibits GA-induced hydrolysis synthesis in barley allural layers. In this aspect, ABA might depress a regular gene or interact with a regulator RNA or protein to inhibit the translation of alpha amylase RNA. So, it has been found that abscisic acid can suppress the seed germination in some respect. Now, the next one, that is the light. It has a pronounced effect on seed germination. Depending on the activity and the response of light, the seeds may be of photoblastic or non-photoblastic. That is, photoblastic seeds are those which respond to light, and non-photoblastic which do not respond to light. Positively photoblastic seeds, if we want to put an example, we must mention about the digitalis purpurea. This is one of the positively very pronounced and very famous positively photoblastic seed. This is known as digitalis purpurea. Digitalis purpurea. This one, positively photoblastic. Positively. And the second one is known as silane armeria. This is another prominent example of negatively, or it's better to say non-photoblastic one. So silane armeria, non-photoblastic plant example. Now, we can also differentiate the positively photoblastic seeds into two types, the first and the second one. The first one acts on a single brief exposure to red light and induces germination. And the second type of the seed, which will only germinate when repeated exposure of light we provide. So the second type is known as photoperiodic, but both these types are phytochrome mediated, and photosensitivity in seeds can be induced by temperature, water stress, and exposure to some other chemicals also. Now, we must study what are the causes of dormancy. The seed dormancy of plants may be due to a combination of various factors. Some of the factors includes about structural restriction or limitation, and some are internal also. Now, the causes of dormancy. First one, impermeability to the seed coat. Second one, mechanically resistant seeds. Third one, seed coat impermeable to oxygen. Fourth one, growth inhibitors. Then rudiment embryo. And the last one, dormant embryo. So these factors regulate the seed dormancy and can express its amount in various doses. First one, impermeability to the seed coat. It has been found at maturity a large number of legumes like alfalfa, cloves, etc., and some aquatic plants, which develop seed coat with hard, thick, and seed coats which are completely impermeable to water. Germination cannot take place until water penetrates through the seed coat. This limitation of water by these hard seeds is influenced by the hygroscopically activated valve in the hilum. That is, this valve controlled the entry and exit of this water into these types of seeds. Then mechanically resistant seed coats. So these seed coats are very tough. In a number of plants, the seed coats are made with some stony cells, which are critical in mechanical limiting and the enlargement of the embryo. Seeds of a number of plants like water plantain, Alisma plantago aquatica of the family Alismaceae, mustards, brassica species, and some other, retain a considerable period of dormancy, as their coats are strong enough to prevent any expansion of the embryo. Then seed coat impermeable to oxygen. This also develop dormancy. The seeds of a number of grasses and some plants of the family Compositae or Astrobici have their seed coat considerably limit the exchange of gases in and out of the seeds. This causes a pronounced dormancy. Then some growth inhibitors. Some chemical compounds are produced in plants and they can suppress the germination process of the seeds. These are known as inhibitors. The inhibitors not only limit the growth but also provide more complex biochemical mechanisms related to the photosensitivity of the seeds. One of the dramatic evidence of the growth inhibitors can be explained by the tomato juice to the other seeds, as well as if we apply this tomato juice to the tomato seeds also. They will provide suppressant to the process of germination. And some other chemical compounds like coumarin, naringenin, this also act as a potent germination inhibitor. So we must know this name of these two compounds. These are coumarin and naringenin. Okay. Then rudiment embryo, reduced or not too much functional. In the development of some of the embryos of some gymnospermous plant like Ginkgo biloba, it has been shown that the embryo does not develop as rapidly as the surrounding tissues. So when the seeds of the plant mature, the embryos are still in an imperfectly or less developed condition. Germination of such seeds is naturally delayed till the embryo is completely developed and it get enough space, enough quantity of nutrients and other supportive factors. So the rudiment embryo, we get this condition in Ginkgo biloba. Next, the dormant embryos. Many mature seeds fail to germinate even all the embryos are completely developed and all the surrounding factors are in a positive attribute or dimension. Because this condition has got nothing to do with the seed coat or the external coverings, etc. etc. Because the embryo is the most crucial or driving force which help in the process of germination. And here the embryo is completely dormant. Like the seeds of apple, peach, pines, etc., germinate only after a considerable period of rest. And this rest period is known as after ripening. We must know the condition is known as after ripening. In case of dormant embryos. Now, there are some methods by which we can break dormancy. What are the methods? First one is known as mechanical, second one as a temperature regulated mechanism, third one is a light, fourth one growth regulators and various other chemicals which can break the seed dormancy. And finally, the exposure to high oxygen concentration. This is also helpful if we want to break the seed dormancy. Firstly, I want to mention about mechanical mechanisms. There is considerable attention we can include for the effectiveness of the mechanical removal, or the process is known as scarification. So, mechanical removal, this is known as scarification. So by this process of scarification, we can remove the seed coat in breaking seed dormancy. This method can be successfully adopted for various plants where the seed coats are considerably hard and tough. Now, temperature. We can apply temperature by two ways. One by using low temperature and one by using alternative temperature. The low temperature is most important. So, by the application of low temperature, when we induce a break dormancy, induce a germination by breaking dormancy, this is known as seed stratification. You must know this term, seed stratification. And it has been found that we can also induce germination by alternating temperature. That is, by low and high, a combination. Now, light. Light can be considered as one of the another important factor for stimulating germination. Its effect is quantitative. Lettuce have shown that red light is the most effective one, which can break dormancy, and the blue and the far-red light has some inhibitory effect for germination. So, the quality of light is important, the nature of light. For lettuce seed, if we induce the red light, it induces germination. If we use the blue or far-red light, it can retard or stop or reduce the process of germination. Now, growth regulators and other chemicals. A number of growth regulators like gibberellin cytokinins have been found to increase the process of germination, already discussed, and some other chemicals which can also induce germination by reducing dormancy include thiourea and potassium nitrate. So, growth regulators, thiourea, and potassium nitrate. Potassium nitrate. This can induce the germination. Now, exposure to high oxygen concentration, this also can help to break the dormancy and increase the rate of germination. And oxygen is mainly associated with the oxidative destruction of various inhibitors. And in this way, they can provide germination. We can get this condition especially in plants of xanthium and some other one. So, today we study the basic features of dormancy, their characters, how they develop, and what are the mechanisms by which we can break the dormancy and induce germination. So, best of luck. Go through your textbook and be prepared for the next class and also for a sudden MCQ test on the all previous classes. Thank you.